Hi everyone, I am Dr. Prabha K, plant pathologist at Directorate of Floriculture Research under ICAR, Indian Council of Agricultural Research. I hope all of you are staying safe, healthy and happy. These days the terms like viruses, symptoms, diagnostics, RT-PCR have become very common due to the pandemic. Virus and virus-like organisms are infecting all living things and plants are no exceptions. And the plant pathologists are the frontline workers who identifies the cause of the diseases in plants and recommends the management strategies. Similar to viruses, in plants there are bacteria-like organisms infects the plant and causes malformations in the plant called phytoplasma. Phytoplasma are obligate parasites which needs a host to complete its complete life cycle, which cannot live outside the host just like viruses. And these phytoplasma are living in the phloem cells of the plant. Phloem cells are the highways in the plant where trucks are carrying sugar to various parts of the plant, and this phytoplasma hijacks these sugar trucks and lives on that. So to live lavishly, it has to keep the plant evergreen. For this, they are interrupting the plant from reaching its reproductive phase, where the flowering of the plant is interrupted and the flowers are converted into leaf-like structures, and this is called phyllodes. You can see here an example where the flowers are converted into leaf-like structures, and this is phyllodes in China aster flowers. Other than phyllodes, during the tug of war between phytoplasma and plant, other malformations also occurs like little leaf, witch's broom, where in witch's broom the plant so many branches arise from a node and looks like a broom. Then yellowing of the entire green plant and stunting of the plant, all these are also occurring. And in case of flower crops, flowers are the economic part where farmer gets the income, and due to phytoplasma infections, great loss occurs to the farmers. So at our institute, we are studying the diversity of phytoplasma infecting the flower crops, flower and ornamental crops, then their infection mechanisms, mode of spread. Then we are also developing diagnostics and management strategies for the phytoplasma diseases. At our institute, we have characterized phytoplasma infecting crops like chrysanthemum, marigold, China aster, and based on the multiple loci in their genomes. And we have developed, we have standardized the PCR based diagnostics for this phytoplasma in these plants. Now we are concentrating on to decipher the mechanism of disease in the plants through transcriptomics, where we are analyzing the differentially expressed genes in the infected plant through next generation sequencing. And these phytoplasmas are vector borne, means to spread from an infected plant to a healthy plant, they need a vector. Insects like causing dengue disease in human beings, we need a vector to transfer, which is mosquito. Same way for phytoplasma, leaf hoppers are the vectors. So we are at our institute, our team comprises of entomologists also. We are trying to identify and characterize the vectors involved in the transmission of phytoplasma as well. Other than phytoplasma, we are also working on the viruses infecting the flower crops. I hope this gives you a picture about the research I am undertaking at my institute. And I thank ASBMB team for motivating to make a presentation before the training session so that we can assess our progress. Thank you all.